So it's an incredible pleasure to introduce Tim and Julia. Julia is really a remarkable person and someone whose story actually has at some level gotten lost within the larger story of her family and her work for Mila, her daughter. She has made it her career's work to really take a step toward not only her own child, but to other children who are affected with rare diseases, and to try to move all of the systems within science and advocacy and medicine to make a difference for rare disease and for patients with rare diseases. And so this particular story begins with Julia, begins with her daughter, and then through an interesting sidelight goes to Tim Yu. This is a great partnership of a patient advocate and a scientist. And Tim is a scientist I know very well because he was a graduate student in my laboratory at UCSF, where he was, the thing I want to say about Tim is that he always wanted to know everything. He was in the neuroscience program. He took all the neuroscience courses. He took all of the biochemistry courses. This was after he had taken 2 years of medical school already. He worked in the lab. He did a residency. He learned everything about neurology. He then went into a lab, learned everything about the developing field of the genomics of human childhood diseases. And that has really placed him to be the person and the partner for Julia, now in his own lab at Children's, to move this field forward in a meaningful and significant way. So I'm going to turn the time over for the two of them, but just express my admiration for both of them and my gratitude for both of them for showing us how things can be done to really start to make a difference in terms of curing, prevent, or managing diseases. Thank you.
It's really an uncommon delight to have the opportunity to be introduced by Corey Bardman, especially as my old mentor, my old PhD advisor and my continued mentor. So it's a special treat. I also want to thank the organizers of this conference for the chance to come and learn more about the remarkable work that's going on in this entire community. And I hope that this opportunity for Julia and I to speak to you is an opportunity to shed an interesting, slightly different light on this type of work from an unusually different angle. Let me please introduce this topic, introduce my partner in this talk, Julia Vitarello. The title of our talk is Mila's Miracle, an individualized ASO therapy for Batten disease. Since this is my first time at this meeting, I wanted to just briefly introduce who I am. I am a neurologist at Boston Children's Hospital and Harvard Medical School. I run a laboratory there where our work up until recently had really focused on the topic shown on this slide, on human brain development, on autism genetics and genomics, on methods for genomic analysis and interpretation, and a dabbling in genomic medicine. What's not on this slide is anything about antisense oligonucleotides, anything about clinical therapeutics. And that's something, a new space that this work has really launched us into recently. In our previous work, working in population genetics, we often worked with large data sets with tens of thousands or hundreds of thousands of genomes or exomes from various patients with different disease cohorts. And instead, our work in the last few years has really pivoted, not because of large data sets, but because of a chance encounter with a single patient, an N of one, that's really allowing us to think about how we bridge discovery and diagnosis on one hand, which has greatly been accelerated in the last decade, with treatments, which continue to be slow and laborious to develop. To really introduce the story, I have the pleasure of introducing Julia Vitarello, who will describe to you how this really all began. And so without further ado, I'll turn it over to Julia.
Thank you, Tim. And thank you to everyone at CZI and the Neurodegeneration Challenge Network as well, just for inviting us to tell Mila's story. I hope that you find it interesting and also find the repercussions afterwards, compelling. So my life as a mother started in a way that was pretty typical to any parent. My first child, Mila, that you see here on the screen, was born very healthy. She had no red flags at any of her pediatric appointments. Her first few years of life were, she was quite advanced verbally and physically. She's a little Colorado kid, so she was hiking and skiing and rock climbing by the time she was 2 years old. And everything seemed to be going perfectly fine. She was learning Italian at 2.5 years old. We brought her to Italy to see her grandparents and our friends, and she was picking up on that. And she continued getting better and better at everything that she did. And I remember at some point, she drew the attention of so many people that strangers would stop her on the street and say, "Oh, you're so cute." And they would say, "What's your name?" And she would say, "My name is Mila Makovitz, and I live in Boulder, Colorado." That was like her tagline. She was very outgoing, really talkative. And so you can imagine that at about 3.5, when she started showing some unusual signs that in the beginning were really just unusual to me. No other doctor or therapist seemed to think that they were quite strange at the time. This advanced child of mine was suddenly tripping and falling. She had bruises all over her legs and arms. She was pulling in objects once in a while, but not all the time. And she would pull them very close to her face. Her character over the period between 3.5 and 5 years old started to change a little bit. Instead of being the center of attention and right in the middle where all the action was, she would be off to the side, kind of reading a book quietly, and all the mayhem of, preschool was happening, and she would be out of that, which seemed very unlike her. She started stuttering, but the stuttering therapist had never heard a stutter like that before. She would get stuck on words, so she would start saying, and mommy was usually the first word. And, at the time it was driving me nuts because Mila would say, "Mommy, mommy, mommy, mommy." And I'm like, why is she saying mommy over and over again? And then I realized that she couldn't get to the second word. And then once she would get to the second, she could keep going, and then she would get stuck again on a word. And, obviously after seeing my daughter being able to hop on one foot around the house and suddenly not be able to hop on one foot and be able to talk perfectly well, and then suddenly wasn't able to get through a sentence, to be able to hike through the snow and probably honestly 4 hours at a time when she was like 2.5 years old. And then suddenly she was tripping at the beginning of the trails and not being able to continue. I was obviously very concerned. And this kind of happened over a period of time between 3.5 and 6. And at some point I had brought her to about 100 different doctors and therapists. I brought her to ophthalmologists, to orthopedic doctors, to neurologists, behavioral specialists, OT and PT, and you name it. I mean, vision therapy, and no one could quite figure out what was going on with her. Around 5 years old, they stopped calling me crazy, and they said, okay, something's going on, but we can't really quite pinpoint it, and she'll probably grow out of it. But it is a little strange. And I kept asking myself, how can my daughter have all of these different problems, all of these potentially neurological problems? They started out as 1 and then 2, but it grew, and there was 4 and then 5 different symptoms. And so when I went to a doctor, the orthopedic doctor, I would also mention that she seemed to have vision problems and that she was stuttering. But no one could really make any sense of this. After lugging my newborn around with me together with my little daughter, who needed to be in a stroller at that point because she was tripping so much, I gave up. And I just decided to bring her into the ER at the hospital closest to us, which was Children's Hospital, Colorado. And I brought her in just thinking there's no way that they can release her. At that point, she was 6 years old, and she was starting to get, much worse. And that turned into a 1-week stay, followed by a genetic panel. And we were told that, she had a very ultra rare variant of an already ultra rare disease, which was Batten Disease. And it was a neurodegenerative condition. And there was no smiles on any doctors' faces. There was no hope. There were no really answers. I asked if there was anything being done for this disease, if there was any treatment, and there really wasn't. There was practically nothing even on Google about Batten CLN7, which is her variant. And I looked over at Mila at that time, and she was radiant. I mean, she was such an outgoing, smiley, happy child, and she was smiling and laughing, and I was being told that she had already fully lost her vision, which I didn't even realize. And she was losing her ability to talk. She was losing her ability to walk. She was having problems choking on food. All of this, and she was still like smiling and laughing all day. And I looked over her and I just thought, this is probably one of the worst diagnoses you could ever have. But if she's fighting, I can fight too. And so I just started calling. I mean, I started reading every article I could. I had never read any article in the New England Journal of Medicine or Science, and I just started reading them. I didn't understand everything. And I started reading about Batten disease, about lysosomal storage diseases, about rare diseases. And I came across names, and I would just reach out to every physician, every scientist, and many parents actually that had fought for their children. And to be honest, at that point, really weren't in time for their own kids, but they had made just Herculean, efforts to raise money for science and advance science in hopes that it'd be for their children. But often, unfortunately, it wasn't. And in those conversations, I got out a few things. I got that parents were fighting and they really encouraged me to fight. And that was hugely helpful because I felt completely alone and I was up against incredible odds. And I also heard, great sadness in the voices of the scientists and physicians that this was one of the worst diseases they'd ever come across. But I heard a little tiny, tiny glimmer of hope of like, it's really early, but there's some reason to believe like we might be able to slow or even stop a neurodegenerative disease, genetic disease. And that's all I needed. I knew it was a long shot, but I just went for it. And I figured I had to start a foundation. That's what everyone had done. So I did. And I started Mila's Miracle Foundation. There was nothing known about Mila's disease except for the gene. And so I went after the gene, and that gene was the only option was gene therapy. You only had to know what the gene was, but you didn't have to really understand, the mechanisms of the disease itself, which we didn't understand at the time and don't understand today fully. And so went after gene therapy. I learned what gene therapy was. I didn't really understand it at first. I started reading more and more. And at some point I realized that it was going to cost $4 million. And I had no idea how I was going to do that. I was not a fundraiser. I was not a scientist, but I was looking at my daughter smiling and laughing. And I just said, I'm just going to figure it out. I don't know how. And the only asset I really had was my daughter's story because people started paying attention to it. And I think one of the reasons they did was because Mila had been so advanced. And so other people that were not part of the rare disease world could see her and think, oh gosh, I lucked out on the genetic lottery. My children are safe and healthy, but my life could have taken that turn. And so more and more people started following Mila's story, and thousands of people eventually, I think in about 55 countries, were donating and asking for updates on her. And, I just started speaking. I started writing. I started writing a blog about my emotions, and I started talking to the press and letting them in my house, like you can see here, which was not what I wanted to be doing at that time. I was doing a lot of crying and not a lot of sleep, playing with my kids by day and making the most of them, and at the same time fighting at night and trying to somehow figure out how to raise $4 million. And along the way, wonderful friends and a company here, Crispin Porter Boguski, which is a big ad agency, just really unbelievably just stood up to help me. And they came into my house when I was so desperate a week after Mila was diagnosed. I just started the foundation, just started raising money, and they came in and they're like, we need to make a really strong video for you because your story is so powerful, but you can't really capture it unless you get a video of it. And thank goodness they did, because it became like a business card for me. I attached it to every single email, and I told Mila's story to anyone and everyone, literally like the person sitting on the plane next to me. And I just figured this is the only way I'm going to do this. And so they captured, I think did a really good job of capturing Mila's story. And I'd like to just show you that very short video now, just to show you that this is what introduced Mila to the world, to scientists, to physicians, to donors, to just followers. So if we could just take a quick look at that video.
Sunshine, my only sunshine. You make me happy when skies are gray.
So our daughter's name is Mila Virginia Makovitz. She was born on November 5th, 2010.
When I think about Mila as an infant, I think about smiles and laughter and health and...
Half an hour after she was born, she was strong enough to hold on to my finger and I could almost pick her up off the bed.
When Mila was a toddler, she was a climber, she was a hiker, she was a runner.
How old are you, Mila? Here's our sugar high daughter.
Slide, slide, slide.
Very physical, always loved to ride a strider,
Nice, bug.
play in the snow, go sledding.
Coming.
We noticed that Mila was getting stuck on her words. She would say, "Mommy, Mommy, I have to, Mommy, I have to," and not be able to continue. And then suddenly, around 3 years old, we started noticing that her feet were slightly unturned and she was walking strangely. And then at 4 years old, we noticed that she pulled books in close. And then around 5 years old, Mila's feet started moving really quickly in this strange pit-pattering way, and then she would fall back on her butt. And every time she stood up, she would fall back and stand up and fall back. You know, we took her around to different doctors who never could really kind of figure out what was going on with her. And then time passes, and then in just 1 month, everything can change. She could no longer see anything. What's brought her so much happiness is playing with her toys and or looking at her books and watching TV and playing with her brother, and suddenly it looked like she could no longer play with anything. It's so hard to see such an outgoing little girl not be able to play with other kids and not be able to play with toys and not be able to watch Elmo anymore. Children with Batten disease are destined to lose their vision, their ability to walk, their ability to talk. They lose their ability to swallow, and they eventually become bedridden, and they don't usually live until much after 10, 11, 12 years old.
It's such a rare disease that all the doctors that you see have never experienced it before.
When Mila was born, it was the happiest day of my life. It gave my life purpose, and I just asked myself all the time, how is it possible that we have such a wonderful life? And then we had our son.
Aslan, he's 2.5, but you know, he doesn't show any sign of being affected, but we know that there is a chance that he is.
The night before we took Mila into the hospital, he looked at me at the dinner table and said, "Mommy, is Mila gonna be okay?" Our beautiful family and life seems like it's slipping away every day. Her smile and her laughter and her spirit is the same. If we want to save Mila's life and the life of other children who are suffering from this horrible disease, we need to raise $4 million right now to get a clinical trial up and running. We want to do everything that we can to stop other families from experiencing the pain that we're going through right now.
You never know, do you, how much I love you. Please, please don't take my sunshine away.
Oh.
That's Mila. And as I started down this path, fighting for her as she was losing her abilities really quickly, I had a problem, which was that in her diagnosis of Batten CLN7, it was actually only a half diagnosis. So it's an autosomal recessive disease. So she needs a mutation from both her mom and her dad, and they could only find one. And we had sent, numerous labs had attempted, and with no luck in finding the second mutation. I was kind of told to just accept the fact that she had this diagnosis, and that probably no lab would be able to either be willing to spend the time to look for this mutation. And even if they did, they wouldn't be able to even figure out whether it was the disease causing mutation, and it might be difficult to interpret. But I had my little son here who was just so radiant and happy, like Mila was at that age. And I had no way to fully test him unless I had both mutations. And I was about to try to embark on this incredible, journey of trying to raise $4 million and start a gene therapy. And I wanted to make sure that the gene I was going after was correct. And so I really pushed through and just said, I really need to find this mutation. And I did my homework and saw that whole genome sequencing was seemed like at the time, like my best shot. And there weren't that many labs 3.5 years ago that were doing this. So I went to social media and I turned to Facebook and I said, please, can anyone help me get into this lab in Harvard that I've heard of that offers whole genome sequencing, but the wait is 5 months and I don't have that time. And I got lots of responses, and everyone kind of tried to help, but no one could really connect me with this lab. But my best friend from college, Amherst College, Jess Flynn, is a doctor in Boston, and she had reposted my Facebook group, my Facebook post to a closed group of physician moms, and that plea of mine ended up on Dr. Yu's wife's lap, within, I don't even know, 24 hours. And the next day he called me and, he said, I saw your plea. I have a lab at Boston Children's Hospital that's working on kind of difficult to find mutations. And I'd like to help you try to find that. So, I'm going to let Dr. Yu tell the rest of that story. He's going to speak in just a minute. But an incredible thing happened, which is that we were going after a gene therapy, and we continued working on that for quite a long time. But Dr. Yu called me back a few months later and said, we figured it out. We found what this missing mutation is, and told me that my son was spared and did not actually carry either mutation, that his life would be Batten free in many ways. And obviously that was such incredible joy after having cried every single night for 5 months, thinking that I would not be a mother and both of my children would die. At the same time, it was a reminder that Mila was losing her abilities really quickly, and that she would. And so, in that same call, Dr. Yu said, I have this great news. We found this, and Aslan has been spared. And then he kind of paused and he took it one step further, something I had never expected. And he said, I have an idea of how I might be able to help. It's kind of a long shot, but go after your gene therapy, but I'm going to do this in parallel. And suddenly, I had 2 potential options for my daughter that had this disease that people told me wasn't going to be able to be understood for at least 10 years and no treatment would be available. A lot happened over that last year, which Dr. Yu will tell you, but to speed up, 1 year, after meeting Dr. Yu, he had designed and tested and had approved by the FDA an antisense oligonucleotide, or an ASO, which was designed for Mila. And we moved our family from Colorado to Boston. And when we got there, we hadn't even had FDA approval. And we got there and within about a week, we got the green light, and Mila basically was about to receive a treatment that was tailored just to her. We learned that she would then become the first person in the world to have a drug customized to just one person. And just as a last little words here before Dr. Yu takes over and tells you kind of the scientific side of this, of what happened in that year, I just wanted to read a very short excerpt. I was writing a blog at the time, and I wrote this blog between Hope's Edge and a Miracle the night before Mila began her treatment. And I think it just captures a little bit of where I was after that really intense year. On the eve of Mila's treatment, I'm filled with many emotions. I've pushed so hard over the past year to get to this point that there was little room left to think or feel. Now I'm here sitting on the hardwood floor of our rental house in Boston with my back against the wall. Mila's treatment is scheduled for 12 hours from now. I hear myself breathing. I feel my chest expanding and collapsing. I look around the room and seem to notice every detail, perhaps because this is the first time I've allowed my eyes to drift in over 1 year. Part of me just wants to hit the fast forward button and have Mila's treatment behind us, but something pulls me back. Deep down, I know that I'm in a sweet spot that I may never be in again, a spot between Hope's Edge and a miracle. Tomorrow, Mila will receive an injection of a new medicine that will go directly to her brain. It's experimental, which means it could be that breakthrough treatment that makes the impossible become possible. Or it could be a valiant attempt that just falls short. She could be the pioneer whose life is saved, or the one whose sacrifice helps those that follow. So I will let Tim take over from there and tell a little bit of what happened leading up to that.
I'm going to backtrack a little bit, and I'll fill in a couple of the details of the story as it evolved from that point. And I recognize that this is a scientific story that's been gotten more than its share of attention in the media, and so some of these details you may be aware of, but I hope that in retelling some of the story that I'll be able to shine some light on some details that might be new to you. First of all, as you know, very unusually, our first connection to Mila was not by a medical referral, but of all things by Facebook. And this is actually the post that my wife shared with me on a Friday evening in 2017, describing how a family in Boulder was on the lookout for a laboratory that could do whole genome sequencing. To be clear, we were not that laboratory. The laboratory that they were looking for was the Lab for Molecular Medicine, our dear colleagues at Harvard Medical School, who operated a clinical laboratory. But we, as a research group, were doing whole genome sequencing at this time faster, actually, than can be typically allowed under typical CLIA guidelines. So we thought that this is something where we ought to be able to help, and we might be a little bit more efficient at it. And it was this initial reach out, thinking that we might be able to help solve an unusual genomic puzzle that led us to an unusual discovery. But before I do that, I have to just point out just a little bit of background about the condition that Julia's daughter had been diagnosed with. She had been given an unfortunate diagnosis of Batten disease. Batten disease is also known as neuronal ceroid lipofuscinosis. In her case, the clinching diagnosis was made by a skin biopsy, which showed these unusual fingerprint-like smudges that show up on electron microscopy. And these fingerprint-like smudges represent the buildup of a substance which is pathologically referred to as ceroid lipofuscin. It's a mixture of lipids and proteins that arrange in a semi-crystalline structure inside the lysosomes of dysfunctional lysosomes of patients with this condition. And this condition is caused, it's a recessive disease. There are 14 different genes associated with it, and they all involve some aspect of lysosomal biology, functioning, trafficking. And in the absence of their proper functioning, cells in the eye and the brain undergo a very, very slow but definitive and progressive neuronal cell death. Mila's particular subtype, CLN7, was a rare subtype. There were 70 cases described in the literature. Very little was known about its function. That continues to be true today. However, clinically, the age of onset was typically around age 3.5, and the symptoms described in a large cohort of patients exactly matched what Mila had experienced and what her family had experienced: vision loss, seizures, motor, and cognitive decline. And very unfortunately for patients with this condition, the age of death is typically around age 11 or soon thereafter. Now, as you all know, we found ourselves in the unusual situation of discovering a path to not only diagnosing her, which was our only goal at the outset, but also to develop a drug customized to her. And we were able to do this in a year. And I'll explain to you the scientific milestones that allowed us to do this work. First was whole genome sequencing. Our offer to the family was to try and solve this genetic puzzle, to do whole genome sequencing of Mila, her mother, her father, and her brother to help clarify his risk for Batten disease. And the initial insight, the key insight, was that on whole genome sequencing, we found, circled in yellow here, a cluster of unusual reads that were in the CLN7 gene here. However, you might note, if you're staring very closely, that this cluster of reads was not within any of the protein coding regions of the gene. It was a deep intronic region, and therefore not detectable via standard clinical sequencing. And when we inspected these more carefully, we found that these were actually chimeric reads. These were reads that matched up to the reference genome. As you know, whole genome sequencing is performed in small bites of about 150 nucleotides at a time. And what we were finding were reads that might contain, for instance, 75 nucleotides that match perfectly to the intronic locus at the CLN7 gene, also known as MFSD8. But then after those 75 nucleotides of perfect matching homology, it diverged into a sequence which was foreign. And on one side, there were foreign sequences appended to the CLN7 gene that consisted of poly T stretches, T, 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 T. On another side, there were a different family of reads that fused to a different foreign sequence, a tandem hexameric repeat, in this case, GGG, AGA, GGG, AGA, over and over and over again. And as far as we could read into the reads, that's all we saw. So there were long stretches of this multimeric sequence on either side. And after a lot of sequence gazing and puzzling over this, we were able finally to deduce that this actually was indicative of an insertion of DNA, in particular the insertion of a specific type of DNA, a specific type of mobile DNA, which is called a retrotransposon. Now, this retrotransposons are jumping pieces of DNA. They are genomic viruses, so to speak, that hop in our genomes, and they are evolutionarily acquired. And they can vary from person to person in the genome because they pick up and hop from time to time. Luckily, this doesn't happen that often, and most of the retrotransposons in our genome have been accumulated over millions of years of evolution. This particular retrotransposon was an SVA retrotransposon that had a hexameric CCC TCT repeat on one side and a poly A repeat on the other side. And these were the exact reverse complement of what we were reading in our genome sequencing data. And SVA retrotransposons are among the youngest of the mobile DNA elements, evolutionarily youngest. They are relatively active, and they can hop around from time to time in the population. Any two people in a given room will have differences in SVAs in probably about 3 to 5 of their SVA sequences. And if they do happen to hop and land in the wrong spot, they have been documented to cause disease. There are about a dozen reports of this in the literature. And so at the time, Mila's case was case number 13. Now, that was our genomic puzzle, except that we had to explain what it was actually doing. Finding this wasn't sufficient. It was a deep intronic sequence, but piecing together what's known about SVAs and the types of sequences that they encode, it turns out SVA sequences encode a lot of splice enhancers and modulators within their repeat regions that we hypothesized and then confirmed experimentally that this mobile DNA element, which had landed in Mila's genome, and it had created a new splice site. It created a new splice site in between exon 6 and 7 where it had landed. And that creation of a new splice site caused splicing from exon 6 into the new novel site, which created a poison exon, an early stop codon that was hit 3 amino acids into this new reading frame. So it was not an open reading frame, but a closed reading frame. And we found this in Mila, evidence of this mutant isoform. We found this actually in mother as well, who was the source of the inherited retrotransposon, and it was absent from father. Thankfully, it was absent from Aslan, Mila's brother, as well. So that was the story. That was what we had set out to do, to solve this genetic puzzle. And by all rights, the story ought to probably have stopped there, except for the fact that we began thinking about this and spurred on by the work of a number of folks, some of whom are at this conference. We began asking ourselves, actually soon after discovering this, can we actually fix this? Right? And on what basis would we actually think that? Well, around this time, this is spring of 2017, we and many other people in the scientific community were reading about this really remarkable success story about an antisense oligonucleotide for spinal muscular atrophy. And we all heard the beautiful talk by Frank Bennett and by Don Cleveland describing how this work came to be, how this class of drugs, antisense oligos, are showing fantastic promise in all sorts of conditions. It's an idea that's been around for about 20 or 30 years, but it's really coming to maturity right now. In 2017, what we had was Spinraza or nusinersen. We had the spinal muscular atrophy story, but that was enough to give us this idea. What is this drug? What are these antisense oligonucleotides? Well, as you heard yesterday, antisense oligonucleotides are synthetic RNAs, nucleic acids. They're slightly modified versions of RNAs with slightly modified nucleotides, modified backbones. And nusinersen or Spinraza in particular is an 18 nucleotide antisense oligonucleotide, and it's given by spinal tap 3 times per year. You heard yesterday about how it binds by traditional Watson-Crick base pairing to a splice regulatory element in the SMN2 gene, and there it restores normal splicing. And you heard yesterday about how it has been shown to have remarkable benefits for infants who were treated early, sufficiently early. And we were reading about this, and we began thinking to ourselves, gosh, our situations are actually really quite similar. And is there any reason why we couldn't make a version of Spinraza for our patient, given the type of mutation that she had? And we laid out the case to ourselves. We tried to convince ourselves that this wouldn't be possible. There surely must be a problem with this idea somewhere. But looking through parallels between SMA and our patient's mutation, it seemed that we were just struck that there were many close parallels. These are both recessive conditions. They both neurodegenerative conditions. SMA was treated with a splice modulating oligonucleotide. We felt that our patient's mutation ought to be treatable using that same type of chemistry, that same type of approach. SMA is a disease of the spinal cord, and so you can access it by giving intrathecal injections. Why couldn't we do that for Mila as well? And could we activate the latent copy of her CLN7 gene, which we must recall was entirely intact. It was a non-coding mutation that was interrupting it, but the protein coding sequences were all there. The question was whether or not we could actually do that. And looking at the background data that supported the SMA intervention, everything seems actually also supportive, that given intrathecally, it's absorbed by spinal cord, but also by brain. You can see it at higher levels in lumbar and thoracic and cervical cord. But when you look at levels of drug, this is what this bar graph is showing, you find drug also in cerebellum pons and importantly in cortex. And the gradient between spinal cord and cortex is only about 3-fold. So it looked like we could get it there. And as you heard yesterday, there's broad neuronal uptake of these oligonucleotides in the absence of a vector. We didn't require a viral vector. We didn't require lipid nanoparticles. We didn't require anything additional that might incur additional risk. So it really seemed as if this ought to be something we should try. Now, the daunting piece, of course, was that SMA, from the concept of treating SMA using a drug like this, there was beautiful, elegant, but painstaking work that had occurred over 10 plus years to make that happen. Adrian Craner, Frank Bennett are responsible for a large chunk of that. And reading their papers was both daunting but also inspiring to see whether or not this path could be replicated. And so pulling all of this together, looking at exactly what they had done and stripping it down to the bare minimum of what we could imagine doing. Knowing, for instance, that we didn't have a mouse model, that we wouldn't have a mouse model. We began crossing things off the list. And we're left with a core set of experiments that we naively thought we ought to be able to do in a couple of months. Of course, with a little bit of help from the path that Frank and Adrian and others had already laid before us. So we devised a plan to design a personalized therapy, and the very simple concept was that we would do a patient-specific screen using cells from Mila, and what we published and what you've heard about is how we used patient-specific cell lines, fibroblasts, lymphoblasts, as a test bed for designing and then validating antisense oligonucleotides to try to block this abnormal splice event and restore normal CLN7 gene function. And this work rested on bioinformatic expertise of our lab. Jinkut Kim and Aubrey Susi designed a series of splice-altering ASOs targeting the splice regulatory sites that were nearby her unusual mutation. It rested on molecular biology experiments done by April, Jay, and Aubrey to screen for splice rescue in patient cell lines. This is an RT-PCR assay detecting normal splice product on top and abnormal shorter splice product on bottom. And what we were able to see is that both products are detectable in patient fibroblasts, but you can bias the production of these different types of products. You can make more of the normal splice product if you add an exon skipping oligonucleotide of our design. And so you can see that in the green circle there. We could flip the ratio and increase the amounts of normal splicing. And we were able to also take this type of experiment and look at EC50 and IC50 curves to compare potency of our laboratory candidates to something which had worked in patients before, to Spinraza. And we found that all ASOs were working with similar potency to established, bona fide, properly developed antisense oligos like Spinraza. And we were able to support this further with transcriptome profiling, looking at more detailed splice junction reads, and to prove to us that we could not only suppress abnormal splicing, but also boost the amount of normal splicing that was occurring with treatment with our oligonucleotide. And then with some very generous collaborators, Joe Mazzulli at Northwestern and his group, we were able to demonstrate functional rescue. This is one of the beautiful pieces of data that Joe's group provided for us, looking at electron micrographs of the cytoplasm of normal cells here on the top left, Mila cells, which showed the accumulation of these abnormal lysosomal storage granules scattered throughout the cytoplasm. And what we saw was that treatment with our oligonucleotide really strikingly diminished the number and the volume of these abnormal lysosomes, these abnormal storage bodies, just after 24 hours of treatment. And this type of evidence, accompanied by other detailed, elegant biochemical assays that they had previously devised and applied to our case, really convinced us that this was looking surprisingly like it was working. All of this, it goes without saying, was done under really tight timelines. We knew that in the course of this typical disease, patients decline over periods of months. And even though we were able to do all of this work, whole genome sequencing, identifying the retrotransposon, thinking about ASO treatments, cell lines, testing these ASOs, we were able to navigate that in about just over 10 months. Despite that aggressiveness on our part, the disease was also aggressively proceeding as well. And during those 10 months, Julia's family described to us how she was continuing to lose neurologic milestones. And we decided we needed to accelerate, and there's plenty of work to be done to take something from proof of concept through manufacturing, testing, formulation, animal tox, and trials. But given the unusual clinical circumstances, we argued that we ought to be able to accelerate this. And by talking to lots of folks in industry, in the FDA with regulatory experience, Art Krieg, the president of the Oligonucleotides Therapeutic Society, Lauren Black, Mary Pendergast, Sarah Goldkind, ex-FDA veterans who advised us on the regulatory routes. We actually devised a plan to tackle these challenges as quickly as possible. And we didn't feel bound by previous historical precedent. We felt bound only by the science, what would be logical to do, not what was typically done. Now, there were lots of challenges. There was no applicable FDA guidance for cases like this. By way of example, making the drug, manufacturing the drug quickly but safely is the most critical step. And for instance, we needed a very, very small batch. We did back of the envelope calculations based on Mila's weight, based on the dosing schedule, based on the potency of our drug. We calculated we only needed 10 grams of oligonucleotide, which is not a small amount for a research lab, but it's a very easily obtainable amount if you just go to IDT. Now, of course, we couldn't just go to IDT to do this. And traditional GMP manufacturers that make pharmaceutical grade drug, they could do it, but they typically made 50 times more than that at a batch. So finding someone who is able to do this with rapid turnaround, with proper sterility, and then to negotiate the quality regulations took a lot of guidance from the FDA, a lot of discussion, a lot of negotiation, and many, many people who stepped up to help us. Here's just a little glimpse of what some of that process was like. It was not always straightforward. For instance, we ended up in an extended legal discussion at one point about what the proper labeling for this product ought to be. And this was not a traditional GMP manufactured drug for a clinical trial. This was made quickly by some really dedicated manufacturing partners with us. But we had an intense discussion about how it should be labeled. We had originally described that the product should be labeled for research use only because we wanted to make it clear this was not a clinically manufactured drug under standard circumstances. But they actually, there was some pushback, and they wanted it to be called not for clinical use, which didn't seem appropriate to us. So we came back and we said, well, I think we need to capture that it's investigational only. But that wasn't satisfactory. They said, well, we should call this research grade. And I said, well, we're getting closer, but why are we beating around the bush here? We know exactly what this is for. We know, you know exactly what we're going to do with this, which is we're going to take it, we're going to inject it into a young girl from Boulder, Colorado named Mila. So why don't we just call it for Mila, or maybe something else, investigational use. Finally, at the end, what we settled on, what we were able to negotiate was this long label here, which barely fit on the vial, but it was what made all the lawyers happy. And I say this not to throw anybody under the bus, or lawyers in particular, but just to point out how challenging this new space was, and how uncomfortable all of this was as we were trying to navigate it. Okay. At the end, though, we did navigate it, and with really, again, fantastic help from TriLink Biotechnologies, Brammer Bio, Craig Dobbs, Richard Snyder, from Charles River Labs, Lauren Black, Julie Duville, and others. We were able to get through manufacturing and animal toxicology in only 3 months. And this was an extremely accelerated timeline, and really relied on many people advising us on what would be appropriate for us to shorten, and we're very grateful for all of that help that we had in doing that. And so we were able to do this, and we were able to get FDA permission to treat our patient on January 19th, 2018. Here's a photograph of the letter that we were waiting for with bated breath. Study may proceed, and it arrived on a Friday afternoon at about 2 o'clock, and I will note that at 5 o'clock, the government actually went into shutdown mode that week. So the FDA pulled out extra stops to get it to us before they incurred a hiatus of unknown duration. So thank you, FDA, for that. And we started our trial on January 31st, 2018. How did we do? Well, so you may have seen reported in the media that this, it's certainly a huge effort on many people's part, and we've seen very, very promising signs, and that's absolutely the case. Now, I do also want to say that this is not a miracle drug, and we've had successes, we've had areas where we feel like we've had failures too. The most important thing is that this was well tolerated. There have been no toxicities to no adverse effects that we can attribute to the drug itself in the now 2+ years that we've been giving it to Mila. And some of our successes have been that especially at the first year, we saw a stabilization of her neuropsychological capabilities. And we saw improvements in her seizures. Where are the places where this drug hasn't appeared to have the effects we could have dreamed for? Her brain volumes, as assessed on serial MRI imaging, have continued to decline, albeit slowly. But we have not yet been able to arrest the shrinkage in volume, which was ongoing for a few years before we started the trial, but that did and has continued as well. With respect to seizures, however, we take great solace in this data, which showed that in the very first year of treatment here, treatment began at the end of January 2018. She was having between 15 and 30 seizures per day that were quite intense, lasting greater than a minute. And with escalating doses applied from January through April or May, we saw a sharp decline in the frequency and the intensity of those seizures. And this was both by parent report, but also on the documented 24-hour video EEGs. And in addition to the decline in seizure frequency, we see that we are going from solid dots to white dots with gray dots in between. What that represents is that the seizure length, the seizure intensity was also being reduced. such that at the start, they were all lasting greater than a minute. And soon by June, none of them were lasting greater than a minute. Almost none of them were lasting greater than a minute. They were typically a few seconds each. And these trends have been stable over the first 2 years, where seizures came down. And at the beginning of year 2, you notice that we gave a few extra doses and decided to increase the dosage amount from 42 to 63 milligrams, because we were seeing that the seizure frequency was still beginning to bounce back up again. The intensity was still low, the duration was still low. So we decided to increase a little bit, and it appeared that then seizures also did continue to further decline. So with that, let me pause there, and all the caveats of an N of 1 study being implicit, I'm going to turn it over to Julia, who can share some of her observations for what this experience was like from their standpoint.
Leading up to Milasen, right in the last 2 months before the drug had been manufactured, you saw some of the incredible hurdles that Dr. Yu and his team were able to overcome in this, very new regulatory path that had never been, kind of followed before. And at that same exact time, from the end of November of 2017, through the beginning of 2018, when she began Milasen, Mila took a really sharp turn downhill. She had not had any seizures that were visible to me, and suddenly she was having more and more and more every day, to the point where she was having up to 30 a day, where I had to pin her down with my full force, and she would have arms and legs, smashing against tables. She was covered in bruises again. And, she was not able to take anything by mouth anymore. And we had to put a G-tube in, and she had to have all of her fluids and her nutrition given to her through G-tube. Her body was starting to slump. She had a decent posture before, but her upper body and her legs and her arms were all starting to kind of give out. And I had every day had her push her stroller very slowly, maybe a few houses down, and her legs were starting to buckle and give out just in the last month before Milasen. So this, race against time was very real. And going into Milasen, Dr. Yu had explained to me that Mila's neurons, some of them had already died. Some of them were in the process of dying and probably couldn't be stopped. And some of them were in the process of dying and they could be stopped. And that actually really helped me, because it helped me set expectations correctly. It was unbelievable that Mila even had this chance, and it looked very promising in the lab, like you just saw. But I also realized Mila had lost a lot and was losing a lot. So by the time she began Milasen, she was having a really, really hard time in every area possible. But in that first year, pretty soon after starting Milasen, we saw truly, a stabilization of her disease for the first year. And we saw pretty amazing improvements. Her seizures, like you saw in that chart, from a parent's point of view, at home, she was pretty much seizure free. We never saw any of those seizures again that were really violent. She had very small ones that lasted a few seconds, and they were down to a couple a day that you would never, if anyone walked in her house, you wouldn't even notice she had seizures. There were a number of days where she had 0. And she started holding her body up really well. She held her upper body up really well. She started eating by mouth again. It was pureed foods, but she was able to eat perfectly by mouth, and she didn't have to receive any nutrition, just water by G-tube. I remember in Boston, there was a really steep set of stairs in our rental house, and Mila had not been able to go up that she had great support from behind at all times with taking steps, but she couldn't lift her feet up to get up the stairs, even with great support from behind. And suddenly I remember there was a day where she lifted her feet up, alternating all the way up the set of stairs. And she actually did that a number of times over that year, not every day, but she did it enough times that I was pretty blown away. And most importantly, my daughter was not, you can see her smile, she was not smiling and laughing as much as before. There's a lot of times I would tell her favorite joke or, read her favorite story, and she always would smile and laugh on cue at certain places in the book or the song. And she wasn't consistently doing that anymore before Milasen. And in that year afterwards, this photo I think is about 10 months after beginning Milasen. And she she was smiling and laughing, not perfectly, not all the time, but a lot more than before. So as you can imagine, I was pretty blown away. I had high expectations, but not this high. And then in years 2 and 3, we're halfway through year 3. So she has been receiving Milasen for 2.5 years. So as in years 2 and 3, I did start noticing that her disease was progressing very, very slowly. It was actually hard to tell until I would look back maybe a year before and notice that she was a little bit worse off compared to the year before. Compared to other children with CLN7, there aren't that many in the world, but the ones that I know, she was definitely doing significantly better. And I'm very, very grateful for the fact that, over the last 2.5 years, Mila's quality of life has been much better than what it would have been. And she's not medical. So we're not in and out of the hospital ever. We don't have any real problems, to the point where she had been on seizure medicine, and I was able to lower that over time. And then very recently she had some new seizure activity coming on, and I was actually able to get her off of some of those seizure meds. And she definitely has myoclonic jerks throughout the day, which is typical of her disease, but significantly less. They're for us as parents, it's really minor. She doesn't have any respiratory problems, and we're not making trips to the ER. All of this said, I would say that I know that Mila had lost a lot going into this. She was still in a place to fight for her when we began Milasen, but she has a very aggressive neurodegenerative disease, and she started receiving Milasen when she was 7 years old, which is a pretty advanced point in the disease. So I don't know what my daughter's future holds. I really don't. But she is losing abilities, and her disease is continuing to progress. And, I ask myself often, I try not to look in the past because it doesn't really help, but I do wonder, if Mila had started receiving Milasen a few years before, and even honestly, even a few months before, where we would be today, because she took a pretty drastic turn in just the few months leading up to Milasen. And I look at photos of her even 3-4 months before Milasen, and she was doing really well. So, time matters in rare disease, especially in neurodegenerative diseases. And so I am, this photo was taken this week. I'm just in a place where I am incredibly grateful that Mila has had an increased quality of life. And we've seen such amazing things from Milasen in the first year. And even in the last 2 years, that it has not stopped the disease, but has greatly slowed it. So I'm very, very thankful. And I'm also thinking a lot right now about the many, many, many parents of rare disease that reach out to me now because of Mila's story, and how many other Milas there are out there. But I'll let Tim talk a little bit about, what happens after Milasen and what implications there could be for other children like her.
I have a couple of slides to reflect a little bit about the case and to think about directions where this experience, I think, is beginning to take us. First, I just want to comment that as a scientist running a basic science laboratory, but also wearing a clinical hat, these are not clean experiments. This is very different than doing bench science. At some point, not everything is tied up in a pretty bow. And at some point, we had to take a look at what we had and ask ourselves, do we have enough to justify a treatment here, to offer both that hope, but also that commitment from the family to engage in this. And I wanted to just reflect a little bit about that. What were the things that led us to think that this was an appropriate decision? And those, I've listed a couple of them right here. First, this was obviously a progressive and devastating disease. And Julia will describe, I think, has described in the past, and will be happy to share with you, that the risks of treatment in a condition, when you're up against a treatment, up against a condition with a natural history of Batten disease, really balance each other out in a significant way. Two, we were working with a monogenic condition with a specific and defined mutation that we had demonstrated was ASO amenable. And I do think that that is important because it was a genetically targeted treatment. And our prior probability going into this, the scientific soundness of it was there in a way that sometimes drugs that you develop, the mechanisms of action are not quite so clear, perhaps they're more complicated here. I think that the prior probability for treatments like gene therapies and genetically targeted therapies like ASOS should be higher, and appropriately so. Third, we were working with a drug platform, Antisense Oligonucleotides, for which there was a lot of data. Not perfect. At the time, there was only a Spinraza that had been given to several thousand patients at that time, but I think still there was 10 plus years of animal data leading into this, and the biodistribution, the pharmacokinetics, the toxicity profile of ASOs were substantially known, continue to be substantially known. And while we are continuing to learn more, that really enabled us to decide to proceed. And last, from the FDA standpoint, this is a rare disease, an N of 1 drug. This mutation that we found in her has not been found in anyone else yet. So it's not a drug to market. From the FDA's perspective, this is individually consentable. There's not a public health hazard here. It's just a matter of whether or not we've appropriately counseled the family as to the risks, the benefits, and all of the unknown unknowns. This work has now been published in the literature about 8 months ago. And it's been received with more than its share of attention. There have been a lot of press coverage of this, and a lot of the concepts that this brings up are the idea of programmable medicines. You know, here we have on the top left nusinersen, which is an 18 nucleotide oligo. And here under it is Milasen, which is 22 nucleotides, exact same chemistry, exact same backbone. How often can this trick be played? Could it be that we could work with drugs like this as a platform to change the sequence, change the targeting, but retain many of the similar properties and thereby expedite the design? And with all of this coverage, there have been, of course, inquiries from hundreds of different directions, from rare disease families, from clinicians, from foundations, all of whom are asking, well, can Milasen be developed for my disease, for my family, for my child? And the question is, what do we tell these families, right? And I will point out that among these hundreds of inquiries, we've identified dozens of patients who, like Mila, have an orphan disease, often a devastating disease. And they have a mutation that looks on paper like it ought to be ASO amenable. So what do we do there? We are really interested in this idea of ASOs as a platform for small n medicine. And thinking through this, we're motivated by a number of things. One, there are orphan diseases, as we all well know, that are too small to attract commercial interest. If you ask various people, it's not clear exactly where that threshold is, 1,000, 500, 200. But there are many, there's a long tail of diseases that have only a few dozen, or sometimes even only a handful of patients who are afflicted by them. And there isn't anyone working on crossing the valley of death in the pharmaceutical lingo to make a drug and bring it to market. Scientifically, these ASOs, developing a platform like this offers opportunities too, to potentially validate a gene target for which you think you have animal data that supports a therapeutic approach, but perhaps in an orphan disease for an N of 1 or N of few that you might be able to consider justifying doing this in a patient. And along the way, you might also be able to answer really critical questions that we don't have current answers to, like therapeutic windows for neurodevelopmental disease. Right? We simply don't know, from some mouse model studies, if many conditions can be intervened upon with meaningful effect. And while, of course, small n experiments have small n limitations, they are better than nothing. And some diseases, these questions might only be addressable in patients. So let's think about that carefully. Regardless, this all has to be done very systematically, very scientifically. And our concept would be to keep the chemistry, the mechanism, the administration route constant, but employ different sequences. It's not as simple as that, but that's the core. And critical here is that from these experiences, if they're aggregated appropriately with appropriate data sharing, then you can build up steadily over time a corpus of information that actually does help de-risk this platform and tell us whether this idea actually has legs. Our paper that was published was accompanied by an editorial by the heads of the FDA, Janet Woodcock and Peter Marks. And there are a number of ethical and regulatory considerations that we and many others are thinking very hard about as we talk about this idea. And individualized therapies, as opposed to therapies for hundreds or thousands, raise a host of regulatory, legal, and ethical questions. But we think it's worth going through these questions and coming to thoughtful answers for them. Regardless of what we think, this work is going to happen. Soon after we publicly spoke about Milasen, there was a case in which actually Don Cleveland and Frank both mentioned this in their talks yesterday. A 20-year-old woman with FUS ALS hit the news last year because she became the recipient of an investigational phase antisense oligonucleotide under an accelerated path very much like the Milasen path. And that has turned into a proper phase 1 trial, but it raised lots of questions about advocacy and the exposure of patients to early stage risk like this. And for our part, we are also continuing to work carefully in this space. We just recently launched a similar small N trial to treat a 2-year-old girl with another neurodegenerative condition called ataxia-telangiectasia. And this is an interesting case where newborn screening in this young girl revealed an immunologic abnormality that led her to be diagnosed with AT. And she had recessive mutations. Both were found at that time, not like Mila's case, but one of them was clearly ASO amenable. It was a gain of splice mutation. And this family, first under the care of UCSF and Stanford, now having moved to Boston Children's, was referred to us through the AT Children's Project, a foundation, which sponsored an effort by us to look and evaluate a number of different AT mutations for treatability using the same paradigm. And we, in the last year and a half, developed an ASO for 2 of these mutations, and then selected this young 2-year-old as the most clinically suitable for a trial, which we launched in January 2020. So more on that in the months to come. Ultimately, the take-home messages are, to put it mildly, the tools for drug development are getting really quite good. And I think it's decades of investment from folks like Frank and his team and Stan Crook at Ionis, and then foundational basic science work by many, many others that have reached this point where biotech companies are able to extend their reaches into rare and rarer conditions, and possibly even physicians or physician scientists might be able to extend their reach as well. I guess that's the question. And it allows us to begin to fantasize about some sorts of futures that could be enabled by further work here, like rapid genetic therapy clinics or newborn genome sequencing that leads you to individualized therapeutics. There's a lot of space between where we are now and there, but the path is not quite as mirage-like as before. So in this space, we've been talking to many folks, and we're working with stakeholders, and especially with the FDA, to discuss these issues. We do think it deserves exploration. We reached out to the FDA and had a meeting with them last May, it feels like an eon ago, to present a white paper where we proposed that we should think about parameters for a pilot where we could see if this type of experience is repeatable. Can it be done carefully? Can we establish safety, tolerability, efficacy in cases like this? And also critically, can it be made accessible? Can this type of work be made more efficient, less costly, both in money and in time and in expertise? And in particular, which of the FDA regulations that we currently employ for large market drugs are appropriate to keep in situations like this? And which might we actually transfer that risk to something like the practice of medicine in the way that transplants or surgeries are performed. Right? And along the way, working with folks like the Oligonucleotide Therapeutics Society, and with the very welcome entry of the NLORM Foundation from Stan Crook and Ionis and Biogen and others into the arena, we need to develop transparent criteria by which these cases are considered for development attempts, and a series of inclusion criteria that really make sense, where we balance scientific, clinical, ethical, systems risk in a way that everyone agrees allows us to go forward. So I'd like to now turn back to Julia and offer her the chance at closing thoughts on the significance of this experience for her and her family and her foundation.
Just some final thoughts here. You know, before Mila was diagnosed with Batten disease, I had no idea that a child could degenerate. I didn't know that anyone that was young and healthy could start losing abilities and die. I didn't know neurodegenerative diseases existed in children. And now, since living this life for the last 3.5 years, and especially since Mila's story has been in the spotlight, I've had so many families reach out to me, and I've learned so much about so many different diseases. And I am shocked. I wake up in the middle of the night sometimes not being able to wrap my head around the fact that there are tens of millions of Milas out there. I don't know where they are, probably behind closed doors crying like I was, and not being able to engage with life, not the parents, the siblings, or anyone, because it's such a difficult life to face physically, emotionally, mentally. And what I've learned is that this concept of a personal medicine or programmable medicine offers something pretty amazing. Right now, we have a pretty much empty toolbox in rare disease. Gene therapy is looking really promising, and I hope that with time, it will be more and more promising. And however, this is, these are going after individual genes, and there are 7,000 plus, different genes that are associated with rare disease. And personal medicines offers, the concept of personal medicines offer a platform that could cut across hundreds or thousands or more of rare diseases. So this offers a potential tool in an otherwise almost empty toolbox. And I think that and I say we because I try my best to capture the voice of so many rare disease parents out there, we deserve the chance right now for this platform to be explored safely but aggressively. We need to make sure that treatments like Mila's get faster and they get better and they get less expensive. Why? Because if they don't, this potential amazing tool will die. We cannot climb this mountain every time. What we put into Milasen, myself and Dr. Yu and his team and about 100 other amazing people that stood up to help us, we can't do that over and over again. So we need to find a way to make this a platform and a really amazing opportunity for families like mine. And with time, we will learn. With time, children will be diagnosed earlier and they will be treated earlier. And, the next Mila will hopefully be able to receive a personal medicine before symptoms even take over. And, who knows what opportunity that offers for that child and that family. So thank you, all for including our story in this meeting. I think it's an important one because attention is often on older people when you think of neurodegenerative diseases. But there is an entire world, unfortunately, of millions, tens of millions of families like mine. So Mila's story may seem amazing and unique, but in some ways it's not. It just shows what's possible now. So thank you very much to everyone at CZI for inviting us to tell this story.
Thank you, Julia. And I just have these closing acknowledgement slides, which I'll leave up if we have any time for questions. But huge teams behind this work and the work that we described. So thank you for your attention. Thank you very much.